Massive thank you for coming on a nice sunny day to listen to talks about AI and biotech and agritech, and also on an England football day, so thanks so much for that as well. Just to give you a little bit of information about kind of what we're doing here. So this is Cambridge AI, hopefully a sort of a series of talks that we'll do for the rest of the year, probably bi-monthly at this point. Just really a community building exercise to kind of, include the kind of wider community of people in Cambridge who are involved in AI or machine learning or data science, just a platform for people to talk about something that's interesting to them or something that they want to show off, or just kind of talk about the work that they're doing, and an opportunity for you guys to kind of network and hear interesting talks as well. So thank you very much for coming. My name's Tom. I work at Robert Walters. We're a recruitment company. So if anyone is looking for a job or anything like that, please speak to me afterwards. But I'm not going to sell to you, so don't worry about that. And I'll leave you in the capable hands of Mark, who's going to compare the evening and fabulous talks from Dominic and Tadas. So thank you very much, and I hope you enjoy.
Very much. So thanks very much, and thanks a lot to Robert Walters for their generous sponsorship of the drinks this evening. So I'm Mark Rogers. I'm from Grounded AI, and we started putting these events together as Tom was saying, as a sort of exercise in community building. We make fact-respecting technology, AI tools and services for science and education. But again, we're not going to go into any of what we do tonight. Instead, we're going to be hearing from a couple of great speakers. So I'd just like to start by thanking our hosts, the Bradfield Centre, James Parton, Mark Walters, Ella Parker, for putting on this event and letting us host it. And it's a very good, very comfortable venue. So I'd like to first start by introducing Tadas, who's our first speaker tonight. Tadas is a physicist by training and an engineer by trade. For over 8 years, he has combined fundamental science with advanced technology in life sciences and drug discovery. At Transition Bio, Tadas combined droplet microfluidics, instrument engineering, and machine learning to address novel therapeutic targets. And he's recently set up his own consultancy, Engivent, and I think it's with your Engivent hat that you're talking tonight. So, Tadas Kartanas, thank you very much.
Okay, can you hear me?
Yes.
Yep.
Okay, excellent. So first of all, I'd like to extend the thanks for the organizers for inviting me here, and thanks for all of you for coming tonight to listen to these talks. I've been coming to Bradfield Center for quite some time, to Cambridge AI, and really enjoy these events where you can meet people from different disciplines and exchange ideas. So I'm quite excited to be speaking tonight and share some of my ideas. So as Mark mentioned, I lately started a consultancy, Engivent, where I'm actually rethinking where I should spend my time in life. So we realized that actually life is so short and fragile at the same time, and we should spend time on things that matter. So at Engivent, my mission is to work with innovators on problems that can make a meaningful impact to the world. The areas that we're starting with initially is lab automation, microengineering, bioprocessing, so anything related to drug discovery and automation, and at the same time we'll have a strand where we're working with smart farming. So that's just a short introduction to Engivent. And today I'd be talking about one way how we can actually drive innovation in drug discovery. And I'd like to start with a slide, which is essentially a representation of how drug discovery works. So we usually start with a lot of information, a lot of new targets, a lot of compounds that we'd like to narrow down. And then essentially the whole process is designed as a funnel where you're narrowing down the information, narrowing down all the facts until you get to one target, one compound that becomes the cure for a disease. Yet this process fails most of the time. It's a funnel, but we still fail to discover drugs, I think, with 90% from preclinical to clinical studies. So it's a lot of waste, you can call, and lots of money spent in unnecessary ways. And how can we fix that? Are we actually reaching a plateau in drug discovery? And I think most of you here are tech people, so you realize that at the moment we're sitting on loads of data. Pharma is sitting on loads of historical data, and the promise is that all of the engineers in this room, together with NVIDIA, will get to new drugs. So that's a new promise, and I'd say in the last 10 years or so, various companies invested heavily in the space. This is a McKinsey report from, I think, a few years ago, where they interviewed, I think, over 100 life science digital leaders. And the main question was, what's the challenge? What are the problems? And surprisingly, low quality data was scoring quite high. So even though we have loads of it, but it's really, really segregated. So there's lots of different sources, the experiments are not well controlled. So then when an engineer is presented with that data set, what can you do with it? You can clean it a bit, but still the results that you can get are questionable value. At the same time, there's a lack of talent, so events like this can hopefully excite some people to join this join this effort. And at the same time, there's lots of proof of concepts that are developed within organizations. But then what happens afterwards, I think there's still a huge gap. So to go beyond proof of concept to full-on adoption, it's actually very few examples where this has been done. And there are a number of biotechs, number of companies that are trying this. And I guess we shall see in the coming decades whether this actually becomes true. So what we can do at the moment is keep working hard. So first we need more data, so we need more process automation, more integration, higher throughput methods for carrying out the experiments. At the same time, we need better data that is more accurate, more reproducible, more standardized, and we also need novel methods which can access new therapeutic modalities. But can we actually turn more data into getting better data? This is a so-called concept lab in a loop, and there are some companies in the world that are utilizing that and have been having proof of concepts. And today my proposal is that, so lab in a loop, just to give a few words, is having your data, building foundation models, and then designing your experiment in a better way to get more data, and then the whole loop starts. So my proposal today is actually use lab on a chip to do that, to close the loop in a more efficient way. What is lab on a chip? So some people have probably heard about this concept. It's sometimes regarded as microfluidics. It's an area of science and engineering which is quite simple, to be honest. It's all of your lab experiments integrated into one chip. So you can imagine, an IC integrated chip where you have all of different functions and amplifiers and all of it that happens. So the same thing can be done for biological fluids. So then it's this sort of experiment is more automated, miniaturized, streamlined. And what are the benefits of that? So it's more accurate to begin with, but at the same time, you can save a lot on the reagent costs. You can make the experiments more repeatable, save space, and most importantly, because I'm a physicist by training, it has unique physics. It has lots of fun experiments that I'll show you in a minute. And the history actually is quite, it reaches back to 1940s. So you could say that the invention of the first transistors may be the genesis of this, because actually how we make these lab-on-chip devices is exactly the same methods as we use to make silicon chips, computing chips. So it's etched silicon in glass, and then combined with various sensors can actually make these devices. I'd say there are a few other notable events that are important to mention. Is Human Genome Project, where we were trying to sequence genome, and that has spearheaded lots of technologies in that space that later on were adopted for different uses. And I guess Illumina is one of the most popular examples that I'll mention in a bit. And the other one is PDMS prototyping. So basically, when you give society a way to prototype, things will happen. Giving Arduinos to people or Raspberry Pis, lowers the boundary to start doing something in that field. And PDMS microfluidics, it's an elastomer that basically allows almost every lab in the university to do this. Now there is probably hundreds of thousands of researchers that have skills in this field, and innovation happens. So as I said, I'm a physicist. I like fun experiments to do. So there are many different examples how this is fun and unique. So you can generate droplets, and today a lot in the talk I'll be talking about this, where essentially in the top left you have water coming from the middle and then oil coming from the sides. And then what you end up is having tiny emulsion droplets of sizes of, say, 100 microns, so 10 times smaller than your hair. And then what you can do in these droplets, you're going to encapsulate different media. You can encapsulate cells, DNA, beads, and do lots of fun things. You can also sort them, so where you can, depending on what's inside the drop, you can direct droplets one way or another, and then you can collect the sample as a whole. You can also use electric fields, electrowetting, where you can move these droplets on the surface and merge them and do various experiments. Lots of fun things to do. So one of the most popular areas in this field is droplet microfluidics, which I said encapsulating aqueous media surrounded by oil. It's minimizing reaction volumes. In pharma, we're using well plates where we have 50, 100 microliters of solution. And then if you shrink that 1,000-fold, you're first saving reagent. At the same time, you can do like millions of experiments with just one small chip. So you can encapsulate things. So you can see in this video cells being encapsulated. You can afterwards inject solution afterward. You can inject solution after they've been generated, so you can make cells live longer. You can also split them, so you can sample solution afterwards and then do some metering during the process of experiment. And finally, you can sort them. So depending what drop it is, so if it's a bright drop, it goes down, and if it's a dark drop, goes up. So for example, if the cell is excreting some antibodies that you're interested in and you label them, you can actually sort those cells and then grow them afterwards. Lots of fun things. And I'd like to finish the introduction to that on some examples that you may or may not have heard about. And I think two of the examples are with DNA sequencing, and actually both of those companies have origins in the UK. So Illumina has commercialized technology initially invented in the University of Cambridge, Solexa, some of you may have heard or maybe have worked there, where essentially sequence DNA by growing the molecule on the surface and then using light to do that. At the same time, Oxford Nanopore is passing small pieces of DNA through a small pore. So it's a highly complex microengineered device that can do this at scale. And both of these devices use very careful fluidic handling, where you have to know exactly when the fluids are going to different locations, when they merge, how they're primed, all of that, combining, of course, with a very complicated assay. And a lot of blood diagnostics and all general diagnostics. So when you go to a clinic, your blood is taken. Some of these tests actually happen on devices like that, handheld, small drop of blood, and gives you some result. So there's some bad examples in this space, and maybe some of you remember Theranos. That's a very bad case, and I think it had pretty bad publicity on this field as a whole. But in general, things work. They just take a long time to happen. So how is Lab on a Chip making an impact to the world? And I wanted to tell this story from really basic start, because I'm aware that maybe here people are not necessarily experts in biology. So cells are very diverse. If you take a human body, how many different cells we have? It's a big number, I can tell. And one thing that someone like a physicist like me who would ponder about, why are the cells different from each other? So we know that cells are made from protein and we have DNA. So how come having the same DNA makes different cells? So actually it turns out there's an intermediate, there's a messenger RNA, which translates certain parts, certain snippets of the DNA into protein. That's the code to make the protein. And the question is, how does that happen? So it turns out that actually the cell is a highly computational device. it's super complicated. There are lots of feedback loops, lots of networks, and the cell can decide what to make depending on its environment, depending on which cycle it's in at that point. And like NVIDIA is saying, we can simulate everything before making our next chips. I think we haven't made a simulation of the cell. It's still too difficult and easier to do experiments with rather than simulate. But maybe we'll come to a point when we can start to simulate cell biology too. At the moment, we know, I think, about 10,000 different cell types that we have discovered, and about, I think, 20,000 genes, if I'm correct, that encode different proteins. And actually can combine these genes in a random way and could get to 10 to 13 different combinations. So really vast numbers. So I think the potential is that there are a lot more cells than we know about. We just know about 10,000, but probably there is millions of them. So the question is, how do you discover them? What are ways to study cells at a single cell level? So if you take any tissue, if it has 100 different cell types, so if you would try to sequence them, you'd get like a mix, and then you'd not be able to get any meaningful information out of it. So this method is encapsulating cells together with beads that have DNA barcode on them. And then with clever assay, you can actually merge all the, you can actually burst the cells open, merge the DNA on the bead, and then use Illumina sequencing for finding what are these individual cells. And then if you do clever bioinformatics, you can actually get to these gene expression profiles, which is, in simple terms, essentially a unique fingerprint for the cell. Now, some of you are machine learning engineers here. If you have lots of data of these fingerprints, multidimensional matrices, and you have a lot of cells, not like 1,000, but 10 million, 100 million, then you have lots of data points. And then if you do clever clustering, there are various ways to do that, and probably a lot of you know more than I do. You can start to see patterns, you can start to see clusters that essentially represent different cell types, and various ways to represent it. So you can project it onto a sphere, and then in this way you can look, are there any clusters that we don't know about yet? And that's maybe a new cell type. And then this sort of approach is discovering cells every week, in lungs, in brain, really expanding area of research. So if you do that for a lot of different cell types, different tissues, you can start forming an atlas. So Human Cell Atlas is a project where essentially we are profiling all different cells that exist in the human body, and really furthering the understanding of not only cell biology but medicine. It's quite exciting. If you go and look inside the cell, there is even more. You go deeper into the forest and you see more trees. How does the cell work? It's as I said, a highly computational device, and we know there are organelles. There is nucleus, there's mitochondria, ribosomes. They have different functions, and it's a highly compartmentalized object. There are a lot of these objects that we've observed for 100 years. There's a tiny blob somewhere within the cell, but we didn't really know what it does. And actually, in the last, I would say, 10 years, there's been expansion in this area of research. And what scientists have found that actually some of these drops are actually liquid, and they appear and disappear. And then if you try to study them, it turns out that they have proteins of different types sort of condensing, crashing out of solution together with nucleic acids. That's actually a way how the cell can feedback and then use as a feedback mechanism to essentially make less proteins or sequester them if it doesn't need them. It's quite exciting. So these objects are called condensates. That's condensomics, various terms that are out there. And essentially this is how a cell can speed up some of the reactions, can get proteins closer to each other to do a certain thing, can also sequester certain proteins if it doesn't want them to be somewhere else. So it's related to various biological functions of the cell, but also, most importantly, dysfunction. And that's where drug discovery can start. And that was the genesis of Transition Bio, where I spent a significant portion of my career. So in this video, you actually see droplets, again, going back to real physical world, droplets that encapsulate different proteins together with nucleic acids, and then we can study these proteins one by one, and these protein clusters one by one, and then try to see what makes a difference. So that's microfluidic device on the left that's encapsulating, again, different proteins, and then we can choose what to mix it with. So microfluidics is really expandable, so we can have 10 components, a bit of RNA, a bit of protein, a bit of that protein, and we can change the composition of them and try to simulate exactly the behavior we have in the cells. So this technology has originated from University of Cambridge, and Transition Bio, we took that to the next level and designed instruments where we can actually screen not one compound, not 10, but actually at 50 to 100,000 compounds now. And we're getting really exciting data, and now we are at a position where we're actually trying to form atlases of different condensates, and then already seeing some behavior and what compounds do what. So I'd like to finish with some opportunities and challenges for Lab on Chip. So those are some examples I gave, but this field actually has enormous potential. So there's a lot of potential for automation, for new ways of measuring things. I think there's this area of untapped. This area that's really untapped is blood biomarker tracking at point of care. And I'm going to finish in a minute. And I think it just requires more people and more people from diverse disciplines to work on this. At the same time, it comes with challenges. So every device takes a lot of time to work out. So for example, Oxford Nanopore, it took them maybe 5 to 10 years to get their flow cell right. So all of these simple things matter. And then if you do them right from the beginning, it essentially spearheads the development of these programs. So that's what I'd like to do at Engivent and enable various companies and innovators to work in that space. So thank you very much for your attention, and I look forward to having discussions afterwards.
Thank you very much, Tadas. Questions. I'm going to hand the microphone to you, so if you wouldn't mind just waiting until you have the microphone before you ask your question.
Two questions. One was the thing of when you're trying to discover different cells and you're doing gene expression to see the different profile, the different signature of the different cells to know. The thing is, depending on the cell status, is going to express different proteins, you are going to have a different signature. So how do you differentiate among they may be the same cells in a different moment, or it may be even cancer? So when it's cancer, it could be anything what you're getting there. So how can you differentiate a cell from its status, or even being a tumor?
Amazing question. You're probably a biologist, but actually, it's quite interesting that because you can do the experiments at scale, you can get data from not 1,000 cells, but millions of cells. You can actually, your data is super noisy, as you suggested, but actually your data quantity becomes quality, and then at some point you still start to see trends, and you can see clusters, and then really machine learning comes into place, how you slice the data and how you identify different cell types. So ideally then you would see different clusters or a continuum within that specific cluster if the cell is changing its gene expression profile during the life cycle.
So then I have another question. You spoke about machine learning. So when you're using machine learning to find the cluster, are you using neuronal networks or another approach that let's say decision trees or any other classifier that is not a neuronal network?
I think typically it's a neural network. But again, I'm not a real expert in this. There's probably someone in the audience who could add to that. But really, various approaches. Are you going to talk about that a bit?
I can talk about that at the end of my talk.
Because this is your company, I was thinking because neuronal networks are very compute intensive, not very ecological, and actually at the end they are not the best in this kind of clustering. You usually need an expert because then you can design better the features that you need to look at, while the clustering is a little bit, you let the classifier decide and it is very compute intensive. So I was thinking in your company, if you are approaching this machine learning via neuronal networks or via any other classifier?
Typically neural nets, but it's an active area of research. So there's really a lot of approaches.
I have more questions.
It's really diverse.
So another direction I see a lot of bio research going in is like really committing to in vivo work, or as close as we get, like maybe like organoids or something, which like by definition are not micro. Maybe could you just like comment on what you see as like the relative benefits of these kind of approaches where you like really isolate cells versus where you measure them in their natural environment?
It's a very, very good question. So actually, organ on chip is another area where microfluidics is at hand. So you can even buy well plates right now where at the bottom you have lots of micro wells within the well, and then they are sort of isolated. But really, the cell environment is critical. You know, what the cell sensors on the surface, it will determine in some sense what it will do. And then I think really controlled environment is crucial later on for introducing different compounds, and then having long-term studies is where automation is crucial.
Can you control at this point in time things like the tension a cell feels? Because I know that is often like crucial in defining shape and stuff like that.
There's some companies in that space. So you can basically have a channel where it becomes wider suddenly, and then the cell just flows and then sort of expands because the flows are laminar. And you can actually see how that cell relaxes and then see what's the time lapse, and then you can infer the surface tension. And in this way, we can actually do cancer diagnostics. And I don't remember the name, but Dino De Carlo was one of the founders of the space, sort of cell biodynamics. What was the area?
Here we go. I kind of missed how your clients exploit the clustering once it's done. Is it they know certain clusters of composition of cells already and they're interested in seeing the new ones, or what? Okay, fine, we have a nice clustering map and so on, but how do they exploit that?
How do they exploit this? Well, I think this is at the moment more towards the fundamental understanding of it.
Right.
But later on, this is an area where you could use it for diagnostics. I think still healthcare systems right now, they are not capable to do this, but you could basically sequence different samples and then identify that you have cancer early on. Just at the moment, if we do that, we still don't have really good ways of dealing with it.
So it's a sort of data reduction method. Like once you have the clusters, then you can match them with statistical studies of particular.
Yeah, but I think that this is really good area to work on in the future. Cancer diagnostics with single cell sequencing, and at the moment the problems are quite basic. It's mainly integration. How do you handle the cells? Who does what at which point? And that's what the industry is facing. But technologically, we can do that already. It's just going to be too expensive, so we don't do that yet.
Very good. Any other questions before we? Oh, lady at the back.
So not a very science question, but more sort of with your consulting business that you set up. Could you tell us just a bit about just an example of the kind of clients you work with, and what stage you're meeting them at, and what journey you're taking them through, and in that, what is the biggest barrier from someone with such an idea to an actual commercial application?
So there's a couple of businesses I'm working with right now where they have a rough idea what they want to do. They want to build like a cell sorter, and essentially they're starting with an idea with some prototypes, but how do they get to the full commercial instrument? That's the path I'm helping them along. So how do you set different milestones in line, and what are the experiments to do, and guiding the team along it, how to grow the team, what are the skill sets needed. But at the same time, there are some other clients where I'm doing purely technical work, where they need to develop certain part of a chip, and they need to make it better, faster, more efficient, fail less. So these are the problems that I'm starting with. But going forward, we'd be really actively looking for businesses in certain areas. And as I said, we are impact driven, so we'll be looking to work on problems that matter. Aging is definitely one area that we'll work in particular early stage dementia detection. I think right now we finally have some treatments for this horrible disease, but not enough longitudinal studies where we can catch it early on. So I think devices like this will be crucial for gathering data that can link our biomarkers and neural cognitive abilities. And essentially, if we catch the disease early on, we can give the treatments, people will live longer. So it's just a matter of when, how, who will do that. So I'd definitely like to contribute to that.
Very good. Final question from somebody?
Thanks very much for the talk. I just have a question about the lab on the loop idea. So is there something about using the data produced on the same lab on a chip to improve the workflow within the same chip, or is this something completely different?
Yes. So the idea behind that is that you get really a vast amount of data, and some of the experiments that start in the drug discovery programs, they are essentially poking in the dark. Just start somewhere with an assay, and then you get results. But then sometimes, a lot of the time, you just really are off target, or the conditions are not perfect. So the idea is that you gather the data, and the next time you do the experiment, starting at the right condition. So you see where the transition happens, where your compound dilution needs to be. So the main benefit of that is that once you start with a new program with a target which is maybe similar but not exactly the same, you start at that point that you already know, and then that saves a lot of resources, from people side to the instrumentation side. So this is a really promising area, and I think then you can expand even more to chemical compounds. What compound libraries you're starting with. So if you have a small set that that affects certain targets, that's what you start with next time. And really the main idea is to have a almost fully autonomous discovery engine, robotic platform that does this for you, and then the scientist sort of just guide it.
Very good. Thank you very much. So we can thank you very much. So our next speaker is Dominic. I'm going to introduce Dominic. Do you mind bringing up Dominic's folder? So Dominic Hall is the co-founder and CTO of Biographica, the target identification platform for agriculture. He completed his Ph.D. in biochemistry at Cambridge, using computational approaches to study 3D genetic regulation. Following his PhD, Dominic worked at Relation Therapeutics, analysing high-throughput CRISPR screens in cancer cell lines. He began to apply ML outside the therapeutics domain. In 2022, he was accepted into the Talent Accelerator and Entrepreneur First, which some of us probably know, and teamed up with Ceci Price to found Biographica with a mission to bring transformative ML advances to the field of plant sciences. He was raised in Devon, and outside of work, he loves surfing. Our next contestant. Thank you very much. Dominic.
Everyone can hear me okay? Yeah? Cool. I'd just like to echo what Tadas says. Thank you for the invite to talk. It's always very nice to chat about what getting up to. And also thank you, Tadas, for what was a really super interesting talk, and going to be a difficult one to follow, but happily for me, I think you covered some of the basic biological concepts, which might give people a bit more of a read into all of the slides I'm about to show. But broadly, I'm just here today to talk about Biographica and Biographica's mission statement, which is the discovery and design of novel crop genetics, and where we think machine learning can contribute to that. So at Biographica, we were really founded on two core beliefs. The first is that food security crisis is one that we should really take seriously, because with rising populations, changing climates, and something happening in Russia, it's only one that's probably going to get worse. And so it's a really urgent one facing our generation. But given my background, I have this strong belief that there's an incredible amount of exciting machine learning happening in the drug discovery and pharmaceutical space, which is directly applicable to problems that are faced in agriculture. And combining those two core beliefs of mine together very, very obviously led to this decision to build one of the world's first machine learning driven discovery platform for crop improvement. Now, what exactly is that, and why do we exist? Tadas showed you a very nice view on the process of finding a new drug. You start from a lot of targets and you go to a very small number of targets and eventually you get a drug. Now I've just gone over, why I think this is important for the agricultural space. But crucially, the agricultural space is going undergoing a change and becoming actually more similar to the pharmaceutical space, in that agriculture is beginning to embrace gene editing as a way to improve crops. Why are we embracing gene editing? It's because it offers the opportunity to get precise and specific edits into plants to bring about beneficial traits that we would all like, such as drought resistance, disease resistance, maybe higher protein content or higher nutrient content. But gene editing faces exactly the same bottleneck that is faced in pharmaceutical industry, which is that efficient discovery of novel gene editing targets is just an incredibly inefficient process. And to highlight just how inefficient it is, I feel like my mic has just gone, but to highlight just how inefficient it is, it's very useful to actually take a look at some of the numbers behind these discovery funnels. And so you can think of R&D pipelines for gene-edited crops having, say, 4 major phases to them. And that's like the target discovery phase. You create gene-edited lines, which are gene-edited crops. You evaluate them in the field, and then you move towards seed production. And we can really obviously see that if we classify these stages by their success rate, then by far and away, target discovery is the one that fails the most. And this is what contributes to a $150 million price tag per successful target that reaches the market. And if you crunch the numbers on that, that means that approximately $120 million of that cost is basically attributable to the fact that we're trying to develop the wrong targets and they're failing later on because of that. And so again, if you crunch the numbers and realize that if you improve the success rate from 5% to, let's say, 30, we're talking about literally hundreds of millions of dollars in savings. And that's exciting not only for the seed companies making these crops, but actually it is more exciting for us all globally as well, because one of the key reasons why gene editing is struggling to be adopted outside of major crops like corn and soybean is that it's really, really expensive. And so many orphan crops, there's simply not the market size to make it an attractive proposition for major research houses to work on it. If you could drop the cost of developing gene-edited crops, many more crops will be gene-edited. And we're already beginning to see this with, for example, Tropic Bioscience licensing the first gene-edited banana in Singapore. Now, this is a big claim, obviously jumping from 5 to 30% success rate. I'm using it in an illustrative manner. But why do we believe there's actually real reason that this could be achieved over the next few decades? And broadly, we think it comes down to 3 major things. So the first is high throughput perturbational screening. And this is of the form that Tadas talked very eloquently about. We're just getting much better at doing biology in a high throughput manner. And the hope is that, and we believe that will just increase the rate of discovery. But obviously, data alone is not just going to give you insights. We actually think one of the things we're really, really excited about is the growth in novel ways to study that data. And I broadly use the term generative machine learning here, but I will define that a bit more later. And then finally, from a technical perspective, one of our big inspirations and the way that it's very natural to conceptualize biology is as a graph or as a network. And over the last kind of 5 years, under the radar of all of the noise of ChatGPT and language modeling, there's been really, really significant advances in our ability to study network biology. And we think it's going to lead to really transformative changes in our ability to
ask biological questions using ML, and start to address some of those questions, such as how should you cluster, or like how should we use data beforehand to inform future data generation. So I'm going to focus in this talk on these last 2 points, and I'll give just a very brief overview of what our platform currently looks like. But I'll be the first to admit this is a horribly complicated slide, and actually this whole talk is just going to be about this bit here, which is around AI-driven functional annotation. So firstly, what is AI-driven functional annotation? I will give a disclaimer here that the results I'm going to show here are about 6 to 9 months old, and in the world of ML, that is ancient. And so I'm ready for your questions afterwards about the various developments that have happened in protein machine learning since we did these studies. But functional annotation is essentially asking the question: if I give you a protein in a cell, a gene product, what does it do? And what questions can we ask about it? We can ask, well, what is its molecular function? How likely is this protein to have adenylate cyclase activity? We can ask, where does the protein localize in a cell? Is it in the chloroplast? Is it in the nucleus? Is it in the cell wall? Does it contribute to the Golgi apparatus? Or we could ask, what biological processes does this protein contribute to? I've given the example here of pyrimidine biosynthesis, but I'll admit that's the only biological process I'm good at pronouncing. They tend to be very, very long. And there's a big problem in that the traditional tools that we use to functionally annotate proteins outside of the lab, they're really, really not very accurate. And what I'm showing on the right here is what's called a precision recall graph. And essentially, it's a way of studying how many times a predictive model, makes false positive predictions, how many times it makes false negative predictions. So this red line here indicates a model that could be used to annotate a protein with some molecular function. And a perfect model would skirt the top-hand side and come down the right-hand side. And what we see is that the traditional tools for annotation achieved an overall score of usually about 0.2, meaning that 80% of the time their predictions were actually false positives or false negatives. And this turns out to be a massive problem in plant sciences because plant science is not as well studied as human biology. Just to illustrate this point, if you go on Uniprot, probably the most popular protein database, and you do a search for corn, which is one of the most globally important crops and studied by all of the major seed research firms and half of the academic labs worldwide, we can see that 784 out of the 143,000 proteins have actually been manually reviewed and experimentally validated. This is less than 1%. It's nearly less than 0.5%. And we don't necessarily think this is a problem, but if you have that number of proteins that exist in an organism and your computational methods for understanding the function of those proteins have an accuracy rate of about 10 to 20%, this is a big problem for your field, because any novel protein you come across, you don't really know what it does, and it really hampers a lot of biological discovery fundamentally. And so this is one of the first modeling problems that we went after in the plant sciences space, because the potential impact, if you could have a computational model which was accurate, is obviously very high. I promise this is probably the most complicated slide on the presentation, but I wanted to give a brief example of a publication that really inspired us for this work, and why we thought it actually might be possible to get a model that is accurate enough at doing functional annotation. Now you'll remember I said at the beginning of the talk we're really inspired by graph-based and network-based methods used in machine learning. That actually extends not just from biological networks, like genes interacting with genes, it actually extends down to even understanding a protein as a network in 3D space. And what this paper showed is that if you conceptualize a protein as a graph or as a network, and if you essentially use some of the protein foundation models, which we've become very successful in biology recently, you can get pretty good at predicting molecular functions of proteins. You can get much better than the traditional tools. This work was built upon in 2023, and then we asked the very, very simple question at Biographica: what if you took these models, obviously sprinkled in a few of the more recent developments, and then said, why don't we fine-tune this for plants? Because these are very general models trained across proteins from like loads and loads of organisms. Why don't we take these models and specifically fine-tune them on plants? And what we found was that you just get this absolutely astonishing domain-specific benefit for training. So this red line was the traditional model that I was showing you before. This green line was the 2021 state of the art, which was what really, really inspired us to go after building these kind of models. And this orange line and blue line represent our kind of like latest models, which we were sort of training around December of this year. And what we found was that not only could we get a lot more accurate than these earlier models, we could actually predict a more diverse range of molecular functions than was previously possible. And these models like directly feed into predictions and commercial projects which we do for customers now. So my lesson number one for any machine learning practitioners is that fine-tuning is always a good idea. Never take a model off the shelf and find the data which will be most relevant to your training procedure. But that's not my only lesson I took from this project, because this was really exciting. We had some machine learning metrics, and we then kind of asked ourselves, like, what next? We've got this ability to, we think, annotate proteins pretty well. What are we going to do with it? And the natural thing was to say, well, there's 143,000 corn proteins which are not manually reviewed, and those numbers get replicated across a lot of different plant species. Why don't we find examples where this model is confidently predicting a certain molecular function, and that molecular function is basically not present in the public domain? So that's what we did, and it led to basically one of our first validated discoveries of a target which we're really, really excited about, and we're continuing to pursue. So ABR1 was a protein in Arabidopsis, which is kind of a model organism in the plant sciences, and it's been known for quite a while that it has DNA binding transcription factor activity. So this protein comes along, binds somewhere in DNA, and then regulates other genes from there. We were super happy to see that our model confidently predicted that was the case. But at the same time, the model also, with 95% confidence, said, I'm pretty damn sure this protein is involved in response to water deprivation somehow. And it was a confusing one for us and a worrying one for us, because this is not in any of the public databases, and it can't be found with traditional annotation tools, even though we tried quite a lot. So we were a little bit worried. And it's at this point I'll say the model that we trained was trained with data up until beginning of 2023, and so it was kind of exciting for us to find that actually in late 2023 two papers got published establishing a novel role for this protein in tolerance to drought stress by basically enhancing lateral root formation by plants, and so even though we're not commercially, going after this target, the reason this is exciting for us is because this is one of, thousands of novel predictions made by this model, and we actually think this is kind of just scratching the surface. This was the obvious model to make, and the number of avenues you could use to improve it is like kind of astonishing. So my second lesson this taught me was that biological evaluation and going into the biology behind what your model is predicting is strictly more interesting than the machine learning evaluation, because although we technically did know these numbers, they don't tell you a huge amount, and actually it's only by really going into the predictions that a model is making that you get some of the biological insight, which leads to actual value generation for us and for customers. So I'll just finish off with talking a bit about what's next, and we're done. We finished. We completed functional annotation. Of course, that's not the case because functional annotation is an incredibly complex task, which is actually context specific. And one of the key ways in which our models, still struggle, and actually all the functional annotation models that we know still struggle, is in prediction of biological processes. So that was the third of the categories which I showed you back when I was explaining functional annotation. Compared to our accuracy at, say, predicting enzyme commission numbers or molecular functions, like we really do a bad job at biological processes. And if you start to think for a bit about why this is, understanding what biological processes a protein contributes to doesn't just involve understanding that protein, it involves understanding the conditions in which that protein is operating, it involves understanding the other proteins that protein is interacting with, it involves understanding the abundance of that protein within all of the cells that you actually care about. There's so many things that are extrinsic to the protein itself that lead to a good prediction. So you can kind of, at a very high level, see our, functional annotation model going sort of from protein sequence and then, making some annotations about this protein's function. But in reality, where we're going, and I think where the entire sort of machine learning in biology field is going, is saying, how do we make those predictions contextual? And so one kind of context might be you ask about both the protein sequence, but also where is this protein expressed? We were talking about single cell RNA sequencing earlier. How is this protein expressed relative to the proteins that it interacts with? How does this protein contribute to other molecular pathways happening within the cell? All of these things are going to be relevant to actually really understanding and making predictions about what to do with targets once we understand them. So I'll just finish with a final slide, which is like, how do we think the best way is to go about this? And I said at the beginning, like we really think biology is a graph, biology is a network. I'll show here on the right an example of a very, very simple network of one protein, another protein. They both have an interaction, and they both get annotated with this trait 2. Protein 2 gets annotated with trait 1. And suddenly, this link between protein 1 and trait 1 begins to look pretty likely because they have strong interaction, and a human being would look at this and say, "I think that's relatively likely." Now, this is an incredibly simple example, obviously, but what we're now building and where we think the field is going is in building models that are capable of reasoning over structures like this, because if you look at a real biological network, the chance of doing that human reasoning goes out the window immediately, because a real network, even of this size, starts to have a lot of extra information which you need to take into account. And all of those are the work of employees of mine such as Vicky, who's here, and my general machine learning team. So I'll just finish by saying, if any of that is technically interesting to any of you, we are hiring at the moment. If you're interested in sort of like graph machine learning and bio and agriculture, then come chat to me afterwards.
Fantastic, thanks so much. So I have a question just to kick things off. So if I'm a big seed manufacturer, do you work for me? And if do you work for me exclusively, or how do you commercialize some of these discoveries?
Yeah, I think our big belief as a company is that the agricultural space will begin to mimic some of the structure you see in the pharmaceutical world. And so I guess the analogy, if you're using that analogy, we see ourselves as like an early stage drug discovery company within that space. Our goal is to develop and license IP, which we would then sell to those large seed firms. I guess one thing to say here is I don't think this isn't a perfect analogy, because seed firms have had, and actually the human race has had for thousands of years, whole other ways of improving plants, and that's breeding, and we've been doing it for a long time, and it's its own science. I guess our big goal is to accelerate the development of the gene editing side of things, but that doesn't mean that the breeding side stops. And so if we become a platform that can develop a plethora or an array of targets which are licensable out to companies, then that's probably the best way to go, because then each can focus on their respective strengths.
Thank you.
So I have a question directly related to that. So do you do you see any value of expanding into synthetic biology? So you could actually be quite active in that space and then build your models at the same time, you do it yourself or partner with other companies to actually carry out directed evolution workflows and develop IP in a more holistic manner.
Yeah, I kind of synthetic biology faces some quite different technical challenges to I think what's faced in plant science, or at least at the moment. Often the things I see them interested in are protein design or maybe even promoter design, or they think of small regulatory circuits. And it's an area where I think it's super valuable, but I don't know if it's where we have a unique ability, I think, to add value, essentially. Often the technical problems we face much more constrained, and they say given this fixed set of genes in some organism, that by the way is probably heavily regulated. Can you work out the minimal changes you would need to reverse some phenotype or something? And I guess it's that regulation bit and the fact that it's quite difficult, which means that there's a double-edged sword there, because the regulation makes it hard, but it also means the value, if we can get that right, is very large, and that's probably a lot of the price tag, right, is around the regulation. So if we can get that right, we view that as a more differentiated point for or stance for our companies to take.
Question really about the models. If we look at pharma, companies that started thought they might have 1 or 2 or 5 AI models and they wouldn't be that expensive to train.
Yeah.
But then they start doing fine tuning and the models become narrower and things, and suddenly they're going, well, we have a technology that might work, but I'm not sure we have a business that works if we've got to train 5,000 models and compute time and various other things and apply many of them. Can you just give an idea how your business actually scales in terms of...
100%. My slightly contrarian... I don't know how contrarian... It's contrarian depending on who you talk to. I think a lot of the way that some ML skeptics, shall we say, talk about bio-ML at the moment echoes a lot of the ways I heard ML skeptics talk about linguistics in about 2015. And I think it's because we're still in this very early phase of even having large enough, diverse enough data sets to do interesting machine learning. And more, I think the trend is going to be moving away from bespoke models or even bespoke data collection, but to very untargeted data collection that is focused almost purely on throughput. Because those are the expensive big models, right? I think are the ones that give you relatively informative priors, and it's the fine-tuning of those models that leads to insights and novel capabilities. The example I would give here, two. There's the obvious one of AlphaFold, right? And the insights that's given to the scientific field. It's not something that you need to fine-tune in order to get insights from. But then the second more recent one, it's popped out of Facebook, called Evolutionary Scale. They also do a protein design task. They do need to fine-tune it to specific context, but what they've shown in their paper is that the amount of data needed to, in their case, generate a novel GFP protein is minuscule. I think they did less than 20 experiments, or I don't know the exact number, but it's a very small amount of experimental effort compared to what you would have had to do to find that without a model. All that being said, I fully agree. I don't think models are just they're not going to fix everything, and I don't think people intend it's not like you'll have a model and that'll be your discovery platform done. But I think that the goal should be, in the same way in the language space, we've developed like very, very flexible models with good priors that are able to be fine-tuned to specific use cases. I think that our goal with all of ML in biology, like data collection, model building, all of those things should be towards building flexible enough models that you're not doing redevelopment every single time, and actually you can focus on the meaningful experimental collection. I feel like I've a little bit dodged your question there, but I've done my best.
It's fine. Thank you.
Any other questions? Observations?
I noticed that on one of your slides you showed LSTM models, a sequence model you used.
Yeah.
Have you tried what everybody else is doing at the moment? Like
Oh, that was an
snap
model.
the
That was an
transform-
model, yeah.
Okay.
That was a 2021 model, and you noticed one of the first things we noticed, which is that they're using a sequence model from like 2002, and the slide after that, which was the 2023 improvement, was to basically make the point that we have better models than that now, and yeah, I think I agree with you. I also find LSTM horrible to train anyway, so.
Yeah, makes sense. And I would also argue with your point about like that fine-tuning is always a good idea, because maybe in most of the cases, yes, but
Yeah.
sometimes it may lead to overfitting, for example.
Yeah, I guess.
In my experience with large models, like fine-tuning big models, has not been the case. I guess that's one of the paradoxes which I definitely can't answer, but have certainly read about a lot, is that you should have this because you've got this massive parameter space of your model. You definitely should fine-tune. You definitely should overfit if you give it small amounts of data, but for some reason you don't. Right? I think like I can't make that claim like absolute in all situations. I will definitely be pulled up by someone who tells me that they fine-tuned and got horrible results out of it. But that's one of the features of these like very, very large models, where you get to a point where they're learning generalizable enough priors that the overfitting, or at least I haven't observed it so much.
Can it make it too? Yeah. Thanks.
Thank you. Any last question? Oh, do you want to pass?
Oh no.
So, it was a very nice talk, and I like the part that you're looking at the problems in the worldwide because of the lack of resources. The thing is, when they started 25 years ago to do gene editing in crops, well, there were a big issue with what they were using to target the seeds. But the issue right now is by stimulating only certain types of crops, you are actually killing biodiversity, and biodiversity is actually the motor of evolution. So meaning that if people started to decide to use only the best crops, they will just reduce the biodiversity of the others, meaning that slowly we will just start potentially kill the process of evolution that brought us here. Which are your considerations regarding that?
So I guess I would first make a point that I don't consider gene editing or machine learning to be like a panacea. I don't think it should just be, applied and obviously everyone should breed the same thing. And a lot of very exciting companies are taking exactly the opposite route, which is we should be learning what we can from wild varieties of crops, because you are correct in that, evolution in some ways it's done exploring for us, and we don't want to like stop that process from happening. That being said, I think there's still value, at least in a small number of crops, for developing them to do, as long as we retain diversity in wild seed banks, which like do exist. Like there is value in going after editing as a way to bring about targeted changes without hurting other traits. a good example of this is like in wheat, the rate of recombination along the chromosome is like very, 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 very low, and then at the end it like pops up. Most of the evolution, when anyone's doing breeding, is happening in the ends of the chromosome. And one of the hopes of gene editing is that areas which are actually very, very, very slowly evolving can at least be, probed in isolation, and we can understand the roles of those genes. And I guess the final point I would say is that even if our thesis is completely wrong on gene editing crops, even as a discovery mechanism, I think that gene editing is extremely, extremely useful. And actually, one of the first companies we worked for doesn't do gene editing. They use a method called tilling, which is basically directed mutagenesis, essentially. But you need, even then, to have a target to go after. And I guess the problem fundamentally which we're trying to address isn't just like people need to be better at gene editing, it's like people need to be better at target identification. And then what that target identification gets used for is, up to them. I just think that gene editing happens to be, or gene edited crops happen to be a particularly promising area, we think, for addressing some of the big problems that face us, like nitrogen use efficiency and drought tolerance and stuff like that.
Thank you very much, Dominic. Very great job. We're going to have a hiatus over the summer. We'll be back in the last week in September, so look out for that. And there are drinks now courtesy of our colleague Tom over there. For anyone who wants to stick around, just for long enough to set up the football. And so it's 10 past now, so we've got a little bit of time. And our speakers, I think, will be staying a little bit longer just to chat. So please come up to them and ask any questions that you have managed to ask in the talk. So thanks very much for coming, and look forward to having a drink with you next time. Thank you.